In this video, we will talking about amylase production. So let's begin with it. Amylase production. Amylase can be used to degrade complex polysaccharide such as starch. So it is important to synthesize amylase as it has various application in pharmaceutical industry, brewing industry, food industry, paper industry, cloth industry. Generally, three types of amylase are found such as alpha, beta and gamma amylase. Out of all these, only alpha amylase are found in human. Now we will see the steps involved in amylase production. First step is selection of microorganism. Second, that is selection of raw material. Third, that is fermentation process, and the fourth, that is recovery. We will see each and every step in detail. First step is selection of microorganism. Various type of organism like bacteria, yeast, fungi are used for amylase production. But mostly we used fungi such as Aspergillus niger and Aspergillus oryzae because of certain reason. As they produced extracellular amylase enzyme. Because of mycelial structure, they can use more amount of substrate. As it will use more amount of substrate, ultimately it will produce more amount of product. It secrete acid, so easily degrade complex raw material by the process of acid hydrolysis. Second step is selection of raw material. Fungi cannot grow frequently on the natural media, so for that we provide synthetic or artificial media. Here is the composition of this medium that containing cornstarch, KCl, Na2HPO4, CaCl2 or CaCO3, MgCl2 into 6H2O. Cornstarch will provide the carbon source, KCl maintain the pH, Na2HPO4 that as a trace element for the mycelial growth, CaCl2 or CaCO3 that maintain the pH, MgCl2 into 6H2O trace element that provide the support for the mycelial growth. This medium is the most suitable, or we can say that the most ideal medium for the fungi to produce amylase extracellularly. Third step is fermentation process. In the fermentation process, first of all, we will take the fermenter. In the fermenter, we will add the raw material. Then we will sterilize. After sterilization, we inoculate our culture, that is Aspergillus niger and Aspergillus oryzae. After adding the culture, we will maintain the temperature around 30 to 45 degrees Celsius. We maintain the pH around 7. After 72 hours, we go for recovery. Fourth step is recovery. Now, after 72 hours, what is there in the fermenter? Three things is there. Our amylase that has been produced, that is there. Our synthetic media and mycelia of fungi. So what we do for the recovery step? First of all, we go for filtration process in which fungal mycelia are removed. Then amylase must be separated from the synthetic media by adding ammonium hydroxide. By adding ammonium hydroxide, that will form the precipitate. By filtration, we collect the precipitate. This precipitate contain ammonium hydroxide and amylase. Precipitate are subjected to crystallization at 4 degrees Celsius. After crystallization, we wash the crystal with sterile distilled water, so we get the amylase in a pure form, and this is how the amylase is produced in an industry.